Let's move on to the next speaker. Next speaker of the session is Dr. Marcela Ovando-Tellez. The title of the talk is An Atlas of the Functional Specialization of the Human Brain's White Matter.
Thank you very much for coming. Let's see how it works.
Could you give a technical support for the speaker?
Maybe
Fine?
mouse.
Okay,
Yeah,
good.
perfect.
Thank you.
Sorry. Okay, so I have no disclosures. So today I'm going to present this ongoing project in this creation of the functional atlas of the white matter. So the study of brain and behavior relationship had led to the division of the cortex into different functional fields over the last decades. And the study using, several studies using fMRI have provided very critical insight into how different brain networks support several cognitive functions, process, and behavior. However, these different brain regions or these brain networks actually are not wireless, and they are supported by the white matter of the brain. And when this white matter is disrupted by any lesion, it can lead to cognitive dysfunctions, right? So this white matter can be classified into association, commissural, projection fibers, as probably you already know. And so these fibers connect regions within hemispheres, as is the case of the association fibers, between hemispheres for the commissural, and between the cortex and subcortical regions for the projection fibers. However, what's the function of these different fibers typically can be studied by exploring brain lesions, right? When there is a lesion in specific tracts, we can see which is the dysfunction, the cognitive dysfunction in the patients, and we could make this relationship. However, what is the relationship between these brain connections and cognitive functions? It's still very elusive, and there is the functional division of the white matter that might be missing. And here, this is what we wanted to address with this study. So we took advantage of this data available in the Human Connectome project, and we analyzed this interval of videos between naturalistic videos and resting data. In 130 participants. So today already have been discussed about the advantage of using these kind of naturalistic videos to explore functional connectivity. So for each participant, we took this data and we explored the contribution of the white matter into these different activations at the brain level. So to this aim, we use a method that was developed in the lab, in my current lab, that is called Functionectome, in which it takes the bold fMRI signal and anatomical priors of the white matter, and it projects the signal of the fMRI data, so the bold signal, into the white matter, and it provides a weight by the probability of the connection of these voxels to the rest of the brain. So by using this method, at the end we have a 4D volume that is like a time series with the information of this brain, the contribution of the gray matter to the white matter into the brain activations during the video watching. So after that, we took this data, we ran one sample t-test in order to explore the activations of these different voxels during all the recording. And we use these two different approaches. So the UMAP, in order to reduce the dimension of the data, as you can see here in the left, and then in the right, you can see this is just an example. The clustering algorithm that we use, the HDV scan, where we can identify the different clusters, so the different voxels that have a similar brain activation during the video watching. And so with this analysis for 3 independent analyses for the association fibers, the projection, and the commissural fibers. And we explore two different groups. So the first, the discovery, and then a replication group. And finally, we validate this parcellation by using a third validation group. So here you can see some of the results. So first, here you have the UMAP representation. So here we see the distribution of the voxels. So each point represents a voxel regarding to the pattern of activation during all the moments of the movie. And this is the data for the discovery group, and we can also see here the data for the replication group. So as you can see, the distribution of these two groups are very similar, and in order to explore if these distributions are really similar or not, we calculated the Euclidean distance matrices between each point in the UMAP for each of the groups, and then we correlate these matrices. And what we obtained was a very significant correlation between the distribution of both groups. And this result actually can tell us that the distribution is replicable in an independent group, so we can trust in this finding. So for the next step, we combine the two groups and we run the clustering analysis. So here you can see the HDV scan process. So here in the x-axis, you can see different parameters that we use in order to find the best algorithm for this classification. And in the y-axis, you can see the score that is a density-based clustering validation score that we use in order to select the best clustering algorithm to parcel or to select the clusters in our UMAPs. So this is the data for the association group of fibers. And here you have the one for the commensurate. And for both, we obtain an algorithm with a validation score of 0.5. And for the projection fibers, we did, using the same method, using the same parameters, we didn't find any clustering algorithm that converged in order to classify the different voxels into different group of clusters. So here, we obtain this division of these voxels that cover 67 and 90% of data for the association and commissural data. And finally, we have these parcellations of with 36 clusters for the association fibers and 40 parcels for the commissural fibers. So then, in order to understand how it is represented in the brain, we map back all this information into the brain. And what you can see here in these representations are the two parcels for the association and the commissural fibers, where each color represents a different parcel in this atlas or this parcellation of the brain based on these voxels brain activation patterns. So here you have, I took one example for the associative parcellation. What you can see here we extracted to better understand what's the functional role of these different parcels. For each of the parcels, we extracted the videos, the part of the videos where they were significantly activated. So here you can see in this graph the different time points of the movie, and with arrows, I try to indicate the moments where this parcel was significantly activated. And in the video that you can see here is the concatenation of the different parts of the movie that were significant. So also, to better understand, we wanted to take advantage from previous data, where in this previous study, they assigned the different concepts, the semantic concepts that were related to each of the frame times in the movie. So here in this word cloud, you can see the occurrence of the different concepts in this movie that you see in the slide. So with all this information, with all this analysis, what we can tell is that we can better understand the role of these different parcels. And then, in order to validate this parcellation, we analyze a new group that is the validation group. So we run exactly the same analysis, and what we did, we took the parcels that we identified for the first group, and we explored the brain activations in the validation group for these same parcels. What you can see here is the overlapping of these brain activations for the parcellation group in blue and for the validation group in orange. And then in order to really explore if these variations across time were similar, we ran a correlation between the two brain activities, and we found significant correlation for all the parcels in the association and the commissural parcellations. So in conclusion, for all this preliminary data is that combining the fMRI and the white matter anatomy allow us to link the brain activations to the structure of the neural connections. And it provides a comprehensive and data-driven white matter parcellation that supports cognitive functions. It also can help to better understand the brain connectivity and function, and can offer new insights for the research that explore the functional role and the pathological signs associated with the variation of the white matter, for instance, when there is a lesion. So I want to thank Michel Thibault de Schotten, who is leading this project, also Chris for contributing to this work, and the fundings, of course. And if you want to know more about the work we are currently doing in the lab, please visit the poster of Chris and Anna that are also related to what I presented today, and Anna about the prediction of outcomes after stroke. And also, please visit my poster if you have more questions, and thank you for your attention.
Thank you very much. Any question? Well, still no question in that. I can start. I think you can potentially use different type of fMRI data set to perform different this analysis. So you can try maybe retinopathy data, or you can try resting state data. Do you expect maybe this kind of parcellation scheme can potentially preserve, or it may be very different?
I think by using resting state data, we can have, I don't know if we can have a similar result, probably yes, but yeah, it has been shown that usually the task or using these kind of naturalistic videos can have a better, can activate better the brain networks that support the different functions. So I think we can better capture these dynamics related to the different functions. But I think with resting state data, it's possible, but we haven't explored, we didn't try that.
Okay. There's one question from the floor in the app. So do you think that commissure fiber pose more challenges when developing Atlas?
I think we, from when we compare the view maps from the different, from the replication and the discovery group, this is the one with a lower score in the correlation between the distribution of the voxels. But the one that it was not problematic, but with the projection fibers were the ones that we couldn't identify a specific role for the different fibers. It was all the algorithm converge in only one cluster for all the projection fibers. But it was not so difficult for the commissural or association.
I think the time is, and thank you very much again. Okay, let's move on to the final speaker in this session. Could you switch